Upstate Medical University in Syracuse, New York invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. Today, we'll hear about a promising treatment for type 1 diabetes with Nidheesh Dadheech. His laboratory at Upstate focuses on regenerative therapies for diabetes, including Islet bioengineering. Welcome to the Informed Patient, Dr. Dadheech.
Thank you, Amber. It is really a pleasure to be here today.
Before we get into the work that your lab is doing, can you please take a minute to explain what the pancreas does and how it's involved in diabetes?
Absolutely. Human pancreas is a critical part of a human body organ system. Although it's a small organ, but an incredibly important one constituting a human endocrine system. It sits deep in the upper abdomen, behind the stomach, kind of tucked between the stomach and the spine, and is about the size of your hand, which is like 6 to 10 inches long. It's about 100 grams in weight, and it's shaped like a flattened pear or a tadpole, having a head and the tail. So pancreas has primarily two main functions to do. First of all, it helps in digesting the food that we eat by supplying key digestive enzymes. And the second, it helps in balancing the energy metabolism. And that's where the diabetes comes in. It makes the hormone that controls blood sugar, that provides energy to human body, improve the growth and development of human tissue. So overall, pancreas is a very important organ.
How does it produce insulin?
Inside the human pancreas, there are tiny clusters of cells called islet of Langerhans. It just like a bunch of grapes. Think of them like land mass scattered with a little island all over the place, and the islets are all scattered in the same way throughout the pancreas. Within those islets, there are very specialized cells called beta cells. And these are the cells that make insulin in our body. Insulin is a hormone that allows sugars coming from the food that we eat to move into the bloodstream and into our cells. That's where it is used as a fuel to produce energy. Just like if you imagine if you have a car and then you try to run a car where the fuel enters the main engine, but without an igniter, the fuel cannot produce thirst. Similarly, without adequate insulin, the cells of our body cannot consume glucose and produce enough energy. And that's where the sugar starts building up in the bloodstream, causing diabetes.
So, if I understand, in type 1 diabetes, the body is not making insulin. Do type 1 diabetics not have islets, or are they there, but they're just not working?
In type 1 diabetes, the immune system is a major culprit. The immune system of the body mistakenly attacks and destroys these beta cells within those islets. In simple terms, in type 1 patient, beta cells acquire some genetic misidentity due to which the soldiers of our human body defense mechanism systems, such as T cells or NK cells, they misidentify these beta cells, target them specifically, and kills them. And therefore, very rapidly, the beta cell pool depletes in patients with type 1 diabetes, and they can no longer produce insulin. People with type 1 diabetes have to take insulin every single day to stay alive, either by injecting needles a couple times a day or having insulin pump that delivers insulin periodically while talking to continuous glucose monitor in real time. Even with the best technologies available, patients can constantly face challenges balancing diet, exercise, stress, insulin doses, and so on. The blood sugars can swing too low, too high, and sometimes severe low blood sugars can be life-threatening. I would say type 1 diabetes is just 24 hours, lifetime condition, and there are no days off, and it's a permanent worrisome life.
Well, what can you tell us about regenerating islets? Where do things stand with our ability to do that?
Islet regeneration is an approach to either preserve, create, or even replenish the lost beta cells by supplying and sourcing new islets. In recent time, beta cell replacement therapy from alternative cell sources have been shown immense promise in replenishing the lost beta cell. And as I mentioned that in type 1 diabetes, the problem arises with the insulin producing cells and that those are gone. If we can somehow replace or regenerate these lost cells or alternatively protect them from immune attack, we can restore natural insulin production. One of the important advancements made in this direction was the Edmonton Protocol for Islet Transplantation by Dr. James Shapiro at the University of Alberta in Canada in 2000. And the protocol is a cell replacement approach where human islets are sourced from a deceased donor and they are isolated from a cadaveric pancreas. These donor islets are transplanted in a matched recipient for treating the brittle type 1 diabetes. Now, since then, over 26 years, islet transplantation approach has been a clinical standard intervention for those who experience uncontrolled hypoglycemic unawareness. In simple way, the goal is to give body back its own natural way of glucose control. But by the way, I just wanted to mention, so before joining Upstate recently, I also worked with the University of Alberta with Dr. James Shapiro and his group in the clinical islet program for over 9 years. And I witnessed the success of islet transplantation and the potential of this protocol to type 1 diabetic patients. And I worked with Dr. Shapiro on how to improve the outcomes with the next generation therapies, such as stem cell therapies.
Well, when you were describing the islet cell transplantation, if I understood correctly, these are taken from deceased donors. The pancreas is taken from people who have died. Is that how this is planned to move forward, or will you be able to generate them from people who are still alive?
Traditionally, you're right that the islets came from deceased organ donor, the cadaveric pancreas, that is used for isolating donor islets. These islets are used in clinic for islet transplantation purpose. But in my lab, we are developing islets from another technology called human-induced pluripotent stem cells, or iPSC approach. These are the cells that can be reprogrammed from patient-own cells, such as blood cells or skin cells or fat cells. And we can now turn them on to become insulin-producing cells. The exciting part of this technology that we are trying to develop in my lab and have been working for many, many years is that we can scale it up now. We can make the renewable sources to produce unlimited supply of these islet cells, possibly even from patients' own cell that may not require the powerful immunosuppressive medications. So it really opens the door to treat many, many more patients that we can ever think of.
This is Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Nidheesh Dadheech, an assistant professor of medicine at Upstate. Now, which patients is this meant for? Is it for anyone with type 1 diabetes, or are there specific criteria?
Right now, in clinical trials, only the patients with type 1 diabetes have been enrolled, and there are some trials that are currently ongoing. There is only one report from China where a patient with type 2 diabetes has been enrolled. But primarily, the therapy is so far being meant for patients with type 1 diabetes. It can also be extended to type 2 diabetes, but more investigational evidence is required and more trials need to be done in order to make this more suitable and applicable to type 2 diabetes. So we really have to go a long way towards treating type 2, but primarily we have clinical evidence that the therapy works great in type 1 patients at least.
Is this available to patients at Upstate yet, or is it in a clinical trial somewhere that people could enroll in?
Not at the moment. It is not a standard therapy at Upstate. Only Vertex Pharmaceutical can do it. Vertex is a pharmaceutical company that has a therapy called stem cell-derived islet product called VX-880 or Zamilacel. Similar to the native islet pancreas, they can also produce islets from stem cell sources. And Zamilacel originally came from Dr. Doug Melton's lab in Harvard Medicine, and later is acquired by Vertex Pharma. In clinical trials, Zamilacel shown promise in 12 patients that has been dosed so far with these stem cell islet with severe type 1 diabetes, and all 12 of them have achieved insulin independence. So this is a major proof of concept in human with stem cell islet, particularly with type 1 diabetes. But at Upstate, we are initiating a new program for personalized stem cell islet therapy with my joining as the Director of Translation Biomedical Program and as an inaugural Director of Advanced Cell Therapy. My lab is bioengineering these clinical grade islets in large scale using a commercially viable technology called vertical via bioreactors in our in-house GMP unit, Biospherics. I have developed protocols that revolves around mass manufacturing these clinical grade products, and we are working to optimize this mass production pipeline at Upstate. Our goal at SUNY is really to initiate a clinical trial to bring this cutting edge technology closer to the patient than ever.
So let me ask you how this works, like from a patient's point of view. How is the transplant done? Is this a surgical operation or is it a simple injection? How would they get the transplanted islets?
At present, islet transplantation procedure is performed in patients by infusing cadaveric donor islets in the liver. From the patient's perspective, this procedure is relatively minimally invasive. With just a radiological guidance, surgeons can conduct the procedure and infuse islet in patient's liver or the vein using a guided catheter. And without a major surgical procedure, the patient can go home typically within a day of hospitalization in most cases. But for stem cell islet therapy, similar to liver, other alternative transplant approaches have also been explored and they're currently under investigation. One key challenge with stem cell islet transplantation from a patient's perspective is the safety concern. Often, if the product is not very pure and have some issues, it can create a safety concerns. And that's why liver may not be a perfect site. So within my own team and one of our colleagues, Dr. Tunjay Delibasi, who is an MD endocrinologist and an expert in islet transplantation, he's focused in this area. Our lab collectively trying to investigate and model new transplantation sites, aiming for improved function and transplantation outcomes of stem cell islet product that we make. One of the key approach that Dr. Dalibasi is focused in transplanting these stem cell islets under a new site, a muscle sheath. We have exciting early clinical data from my studies demonstrating a huge promise in improving islet function with a rapid diabetes reversal for over 8 months in animals under muscle sheath transplantation. So I'm very excited to see the progress, and I think we have potential to deliver something new and alternative site for stem cell islet in clinical applications.
So, how soon would a patient notice a difference after they undergo a transplant?
If the islet cells are transplanted and they function well, the blood sugars control can begin improving within weeks of transplantation. So some patients can reduce their insulin requirement gradually. Some others can achieve full insulin dependence within 3 to 4 months post infusion. Most patients do require immunosuppressive medications with this transplantation to evade immune attack. This is an active area of research, but reducing the elimination of the need for lifelong immunosuppression is one of the biggest goals in the field, including the pioneer work that we were trying to do in my lab. In my lab, we are developing engineered islets, which is safe to immune system, and we engineer them from patient blood stem cells as they become more likely functional and able to handle the immunity without the use of immune suppression drugs. And at least in the case of severe insulin type 2 diabetes, where autoimmunity is not the major culprit, one of the key focus area in my lab, that's where we are looking. So overall, I think it creates a lot of difference in patients and disease management. And within no time, if it's successfully conducted, if we can do it right, patient can start experiencing a major difference and complete remission of diabetes within months.
Any listener who has type 1 diabetes and thinks they'd like to learn more about this, is this something where you would ask their primary care physician to refer them to the Joslin Diabetes Center at Upstate to learn more?
The patient can come to us at Upstate, the Institute of Human Performance. We have an advanced cell therapy unit. That's where I am located, and I can really explain the technology and give them a real sense of understanding how things work, what kind of cells are we making. So we can give a lot of information about the manufacturing side of it. But at the Joslin Diabetes Center, we have some of my colleagues that work with me. They can provide a lot of information about the medical interventions and how it's going to impact medically and clinically, what are the benefits and how these procedures will be conducted, what patients are experiencing or they're going to experience post-implantation. So all the challenges, benefits, potential applications can be explained to these patients prior to the transplantation. We have a very strong clinical team that works directly with patients and provide enough information that helps them to decide whether they really have to go for this procedure at CME Upstate. I welcome all patients and families. If anyone would like to get more information, we're happy to provide that.
Do you have patients that are getting these transplants right now?
We are currently in a process of recruiting the patients, not for the transplantation and the clinical therapy at the moment, but for investigational studies to make sure that the cells are right, make sure that the process is optimal, and we can generate these cells in our facility over here. Once the process is ready, we will apply a package called IND, Investigational Drug Report, to FDA, getting a permission to implant these cells in patients. And that's where the clinical trial starts. So it potentially may take 2 to 3 years timeline from now to reach the point where we can have first patient in and we can dose these cells into those patients. So we're working on that, but I think that's the ultimate goal.
Well, this is very promising. I thank you so much for making time to tell us about it.
Yeah, likewise. I'm very pleased to be here today.
My guest has been Nidheesh Dadheech, an assistant professor of medicine at Upstate, who investigates regenerative therapies for diabetes, including Islet bioengineering. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.